And our next speaker is Christopher Leonard from Utah.
All right. Hello, all. My name is Chris Leonard, and I am a second-year graduate student in Nitin Fudnis's lab at the University of Utah. And today I'm going to be telling you two stories about distorting X chromosomes in two species of Drosophila. First, I'd like to thank our collaborators Zach Fuller and Steve Schaefer, without whom this story would not have been possible. To start, I want to begin with something simple, to remind you of Mendelian inheritance. If you have two competing alleles, when they are produced in meiotic products, these should be present at equal ratios, and in progeny, those should also be equal ratios as well. Today, I'll be telling you about a type of chromosome which actually distorts this phenomenon, so that if you have a driving X chromosome in a male such as this, that now his sperm will be primarily X-bearing, and this will be reflected in his progeny as an overabundance of females. Obviously, things like this can have huge implications for population biology, and I'll tell you now that this is not a niche phenomenon. These are found across a wide swath of eukaryotes and can be at very high frequencies in populations. And so these have really important implications for how we think about evolution and can explain phenomena like the rapid evolution of meiotic genes, meiotic sex chromosome inactivation, and the evolution of barriers to gene flow between species. These have now been implicated many times in the development of hybrid incompatibilities, including by work from our own lab in two subspecies of Drosophila pseudoobscura, and this has really pointed to their role in actually creating these barriers through meiotic competition. But what I'll be telling you about today is actually kind of a counterexample to that, where instead we found that distortion has enabled then gene flow between two species. We still have some essential questions about distorters kind of broadly. First, again, further clarifying how distortion is interacting with these evolutionary phenomenon. Secondly, we have a really poor understanding of what kinds of genes cause distortion across taxa. And lastly, we have an even poorer understanding of what the molecular mechanisms for distortion actually are. And these seem like fairly straightforward questions. In a lot of cases, these are things that people have been interacting with for almost 100 years at this point. And the problem is that these loci are found in inversions. This is how I'll be representing a chromosome for the remainder of the talk, with both a right and a left arm, and then here a centromere in the middle. And then these parentheses on either side then represent an inversion. These inversions have the effect of locking regions of a distorting chromosome away from recombination with a standard chromosome and create large linkage groups, which are probably beneficial to distorters, but essentially cripple standard recombination mapping for finding these loci. And so these have really been a thorn in our side for coming to a genetic understanding of distortion. The primary goal of my thesis across the rest of grad school is actually to find ways to genetically circumvent these problems, and specifically by reinverting a chromosome so that we can enable recombination mapping. And that's not what I'm going to be telling you about today. Instead, what I'll be telling you about is how we've used inversions to uncover a really surprising evolutionary history of two distorting chromosomes. Oh, sorry, these are the single recombination events here. To study this, we are using two distorters which kind of hold a hallowed place in the segregation distortion field. These are distorters in Drosophila pseudoobscura and Prisimilis, both characterized by Sturdivant and Dobzhansky back in the 30s. And they're really similar at the face. Both of them are distorting X chromosomes and cause destruction of the Y chromosome, and almost completely distort, sometimes even like 100% females. These occur at very high frequencies in the population and curiously are quite unsuppressed. And so based on these kind of laundry list of characteristics, at the surface level appear very similar, but actually have very different evolutionary histories, as I'll tell you through the remainder of the talk. And this starts with their chromosomal inversions. Drosophila pseudoobscura SR, or sex ratio, these are distorting chromosomes, contains 3 large non-overlapping inversions on the right arm of the X chromosome compared to its standard chromosome, while Prisimilis has just 1. So there's an inversion between the SR and standard chromosome of the same species, but you'll notice there's a strange collinearity in this diagram between this distorting chromosome of Prisimilis and the standard chromosome of pseudoobscura, and I'll get back to that. To study these in the depth that we need to, the first thing that we need to do is actually collect these strains from the field, and this is work, and here's our outgroup, I'm sorry about that. Is actually to collect these strains from the field. And this is hard work that has been done by Randy Young, who is a former member at our lab and now at University of Wisconsin in Madison. And she found all these strains in the wild, brought them back to lab, isolated them, purified them, and then we sequenced them and aligned these reads back to the pseudoobscura reference genome. So now we have a genomic depiction of what these distorters look like. I'd like to begin today by telling you about the story of Drosophila prisimilis. And I want to focus first on this strange collinearity. This is first noted by Dobzhansky back in the 40s, but I think the first question that comes to mind is, how exact is this collinearity? Are these chromosomes entirely collinear, and does this extend down to the molecular level? I was able to amplify across this region in both pseudoobscura standard and the Persimilis SR chromosomes, and found what others have found before me, this series of repeats which are present throughout the pseudoobscura genome and Persimilis genome as well, which are called Leviathan repeats and were first characterized from work from Mohammed Noor's lab back in 2007. These repeats are present at the breakpoints but are flanked by unique sequences which localize to this specific region of the chromosome. At a gross level, these were similar between pseudoobscura and Persimilis, and when I zoom in just on this breakpoint here, we actually found that these two chromosomes were collinear to the base pair. So this is really surprising because these are across species, and an inversion exists in Persimilis between a distorting and non-distorting chromosome. And so we wanted to ask, are these chromosomes more related to each other than the two Persimilis chromosomes are related to each other? And to do this, this is work done by Zach Fuller, we performed a sliding window analysis across the entire genome. And within those windows, collected sequence from both of the Persimilis chromosomes, the pseudoobscura standard chromosome, and Drosophila miranda as an outgroup. Using these sequences, we then constructed a tree and then plotted these on the chromosome. And each one of these bars then represents a window where we find what I'm calling the species phylogeny, where the standard and distorting chromosomes from Persimilis group together. On the bottom is what I'm calling the structural phylogeny, where we've grouped together those two perfectly collinear chromosomes. And then in the middle here is the standard phylogeny, where the two standard chromosomes are grouped together. And so you can see that on chromosome 3 here, which has nothing to do with distortion, we find that the two Persimilis chromosomes are closely related. However, the story changes when we look at the right arm of the X chromosome. While most of this arm still maintains the species phylogeny, where the two Persimilis chromosomes are grouped together, there are two large regions where we actually find that the structural phylogeny holds more true. So these are regions where the Persimilis distorting chromosome looks like its cross-species standard chromosome instead of its own. And curiously, these are exactly at the proximal and distal breakpoints of this inversion. And so this is really two strange phenomenon. One, we see this incredible collinearity between both of these chromosomes, and this phylogenetic similarity precisely at those breakpoints. And so we've come up with the following model to explain this. Pseudoobscura and Persimilis diverge from each other anywhere from 1.5 to 1 million years ago. We know that they can form hybrids in the wild, and so we probably see examples of gene flow constantly. However, through selective pressures, because most of these introgressions will be maladaptive, will continue to be bounced off the Persimilis population. However, when one of these introgressions acquires drive, then this would found the distorting chromosome in Persimilis, and then through continued recombination between now the two Persimilis chromosomes, we would come to the Persimilis SR chromosome that we're at today. And so this explains why we see the collinearity between these two chromosomes, because they're one and the same. The distorting chromosome in Persimilis is in fact an old pseudoobscura chromosome that's persisted across species. What this looks like at the genetic level is that when these come together to form an F1 hybrid, they'll enter the population, and through single recombination events, quickly purge genetic material outside of the inversion. And then through double crossovers and gene conversion, we'll also purge pseudoobscura material that's maladaptive within the inversions. However, there are a few regions in the genome where this can't happen, and this is exactly at those inversion breakpoints. These inversion breakpoints entirely suppress recombination, and so you can't see these phenomenon. When we zoom into this a little bit more closely, you'll find that this looks strangely reminiscent of our pattern in the phylogeny, where we see both of the retention of pseudoobscura material in the distorting chromosome at each one of these breakpoints. And so with this model, we can successfully explain the strange collinearity across these species bounds, the phylogenetic pattern that we've observed. And from this, we can conclude that drive actually may have circumvented these gene flow barriers, which it itself has been implicated in creating. But this is a tale of two systems. And again, I told you at the beginning of the talk, these are very different from one another. Because the goal of my project is actually to genetically map distortion, we wanted to carefully have a good genetic depiction of what the breakpoints of all these inversions look like. And so that's what we did first in pseudoobscura. We were able to map 4 of the 6 breakpoints here to the molecular level, and we found some interesting things here. First, some of them are perfect. We don't see any new insertions or deletions at the breakpoint. Some of them have small inversions. Others are associated with repeated elements present throughout the genome. In this case, this breakpoint actually perfectly bisects one of them. And then some of them have a huge accumulation of novel material, and these are primarily repeats. The important thing to take away from this is that none of these actually disrupt known genes, and there's no obvious chimeric genes that are formed as a result of these inversions. So it's probably not inversions themselves which are causing distortion. With the more precise breakpoints in hand, we wanted to ask if the SR and standard chromosomes in pseudoobscura were divergent from one another. And to do this, we relied on FST as a measure of differentiation, and we've predicted the following pattern. On the left arm of the X chromosome, which is left of this dotted line, because the recombination can happen freely between these non-inverted regions, we should see low differentiation across this region. But that in places where this recombination is restricted, we should see elevated differentiation between a distorting and a non-distorting chromosome. What we found, however, and this is work done by Zach Fuller again, is that while the expectation for the left arm of the chromosome is true, where we see this low differentiation between the chromosomes, and we also see this high differentiation in regions that are inverted, this high FST actually persists outside of regions that are inverted with one another to places that are actually collinear between a distorting and non-distorting chromosome. And this is really surprising to us because recombination should be able to happen here. We also wanted to see if these chromosomes were in linkage disequilibrium across these same regions, and we also find that to be true. So these are multiple strains of standard and SR chromosomes, and I think you can appreciate, based on the light and dark allele differences, that there are huge blocks of within our markers where we see this pattern of linkage disequilibrium between standard strains and SR strains. And again, these markers are placed across the length of the X chromosome in places that are both within and outside of inversions. This pattern is not completely unusual. Some work from Kelly Dyer and her colleagues a few years ago found a distorting system in Drosophila recens where there is also this elevated FST even in regions that were collinear. The difference between her system and our system, though, is that her system was an extremely young distorter, about 5,000 years, and these patterns were associated with selective sweep. It's been known for about 20 years now that the distorting chromosome in pseudoobscura is actually probably quite a bit older, and so Zach was able to confirm this at the genomic level by, again, doing a sliding window analysis. In blue, I'm showing you the divergence time across species bounds, so between pseudoobscura and persimilis, and in red, across this distortion bound, so between a distorting and non-distorting chromosome. When we look at the ages that come out of this, we find that the species divergence is well placed, about 1.3 million years ago, but that the divergence between a standard and SR chromosome is about 1.1 million years ago. So we see these signatures which are found on this very young distorting chromosome on a much, much older chromosome. Now, we don't really know what's happening here yet, and so I would love to hear your ideas, but we're also really interested in this scenario. So we can say that pseudoobscura SR is old and highly differentiated. It's probably not associated with the same type of adaptive sweep you'd see in a younger chromosome. And we think that this is probably because all of these inversions behave as one non-recombining unit, although that is still to be tested. So even in this small clade, we found two very different stories from something that looked the same at the surface level. In one case, we found an old chromosome that is highly differentiated from its standard partner, even in regions where recombination should occur. And in the other, we found a really interesting example of how drive actually may have enabled transgression of species borders. I want to kind of come back to inversions here. Although they've been a thorn in our side as geneticists for mapping distortion at the genetic level, in this case, we've actually been able to leverage them, and specifically the recombination block, to come to two very different conclusions about the evolutionary history of these distorters. With that, I'd like to thank the members of my lab who've really made this possible, specifically Randy and Josie. Our collaborators have obviously done a massive amount of work for this project. The other sources of inspiration and criticism from our university, as well as our funding. And with that, I'd love to take questions. Thank you. Okay. Yeah, great, thank you.